Hello and welcome to my channel. So today's topic is glycolysis. Glycolysis also means glucose catabolism, where glyco means glucose and lysis means breakdown. So breakdown of glucose, which comes from diet usually. So in this process we will see that glycolysis is independent of oxygen and it is a major metabolic pathway for microbial fermentation also. And fermentation and respiration, they are the two major strategies which are used for energy conservation in most of the organisms. So this pathway is also called Embden-Meyerhof Parnas pathway. Regardless of whether glucose is fermented or respired, it travels through this pathway, thus it is referred to as the universal pathway of glucose catabolism. So let's start. Glycolysis is divided into two phases, preparatory phase and payoff phase. Preparatory phase is the phase where glucose borrows ATP from the cell, and payoff phase is the phase where this process pays back ATP. So the location of glycolysis is cytoplasm of cells in most body tissues. So in preparatory phase, the first step is as soon as glucose enters inside the cell, we want to trap that glucose. Since glucose is a molecule for which we have transporters. GLUT transporters, they help in transporting glucose inside the cell. Similarly, they can help in export out of the cell also. So with the help of hexokinase enzyme, we convert glucose into glucose 6-phosphate. Hexokinase is a kinase which enzyme which helps in transferring phosphate group from ATP to glucose. So from ATP, phosphate group is transferred to glucose so that it becomes glucose 6-phosphate. And hexokinase is a very non-specific enzyme because hexo means 6 carbon. So it works for all 6 carbon compounds. And glucose 6-phosphate, conversion of glucose to glucose 6-phosphate is a very crucial step because in this way cell can trap glucose because it has attached a big group called phosphate group, and this phosphate group attachment helps in trapping it inside. Now the second step is catalyzed by the enzyme phosphohexose isomerase. It's a intramolecular rearrangement. And this glucose 6-phosphate converts to fructose 6-phosphate. So both are 6 carbon compounds, fructose 6-phosphate also. The third step, fructose 6-phosphate converts to fructose 1,6-bisphosphate. Again, a kinase enzyme catalyzes this reaction called phosphofructokinase. This enzyme also borrows phosphate group from ATP and donates it to fructose 6-phosphate so that it can become fructose 1,6-bisphosphate. And Ng2 plus is the cofactor which is utilized in both the reactions. In the next step, fructose 1,6-bisphosphate, which is a 6 carbon compound, it splits itself into two 3 carbon compounds. The enzyme is called aldolase. So fructose 1,6-bisphosphate converts into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So one 6 carbon compound with the help of aldolase converts into two 3 carbon compounds. Now this glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, they are isomers of each other. So with the help of the enzyme triose phosphate isomerase, they rearrange and dihydroxyacetone phosphate also becomes glyceraldehyde 3-phosphate. And with this, preparatory phase ends, and by now we have already taken two ATP molecules from the cell. Now we have two glyceraldehyde 3-phosphate because dihydroxyacetone phosphate is also converted to glyceraldehyde 3-phosphate. So we will proceed with two glyceraldehyde 3-phosphate in the payoff phase of glycolysis. Now two molecules of glyceraldehyde 3-phosphate in the payoff phase. It's called payoff phase because in this ATP will be given back to the cell. So two molecules of glyceraldehyde 3-phosphate, they convert into 1,3-bisphosphoglycerate. So because at the first carbon a phosphate group is added, so for this 2 NAD plus molecule they convert into 2 NADH plus H plus. So NAD is the coenzyme for the reaction. G3PDH, that is glyceraldehyde 3-phosphate dehydrogenase is the enzyme which catalyzes this conversion, and NAD plus converts into NADH plus H plus. So two hydrogen molecule they are given to the reaction. In the next step, phosphoglycerate kinase catalyzes conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. This reaction is called a substrate level phosphorylation reaction. So it's called so because in this substrate, that is 1,3-bisphosphoglycerate, it donates its phosphate group to ADP molecule. This phosphate group is donated to ADP molecule so that it can become ATP. So since ATP is produced in this reaction with the help of substrates called so. So this 3-phosphoglycerate, two molecules of 3-phosphoglycerate, then they convert into 2-phosphoglycerate, again a molecular rearrangement reaction where phosphoglycerate mutase is the enzyme which catalyzes this conversion. In the next step, two molecules of 2-phosphoglycerate, they convert into phosphoenol pyruvate, two molecules of phosphoenol pyruvate. So in this two water molecule, 2H2O, they move out of the system. And enolase is the enzyme which catalyzes this conversion, 2-phosphoglycerate to phosphoenol pyruvate. And now the last step of glycolysis pathway, that is phosphoenol pyruvate conversion to pyruvate. So phosphoenol pyruvate converts to pyruvate with the help of the enzyme pyruvate kinase, and again it's a kinase, so it takes this is a kinase, so it transfers a phosphate group. So this pyruvate kinase reaction, this is also a substrate level phosphorylation reaction. So this is second substrate level phosphorylation reaction of glycolysis, and there are two substrate level phosphorylation reactions in glycolysis pathway where substrate molecule donates one phosphate group to ADP so that it can become ATP. So phosphoenol pyruvate, the group is donated to ADP so that it can become ATP, and the product pyruvate molecule. So two pyruvates are formed and 4 ATP molecules are produced in this payoff phase reaction. And this is the balance sheet. One glucose molecule, and in the preparatory phase it takes 2 ATP and 2 NAD plus 4 ADP plus 2 PI, and it returns back 2 pyruvate, 2 ADP plus 2 NADH, 2 H plus and 4 ATP plus 2 water molecules. So if we cancel them, it becomes one glucose molecule in a glycolysis reaction produces 2 ATP molecules. So 4 minus 2 becomes 2. It borrows 2 and it gives back 4, so it becomes 2 ATP. So one glucose molecule produces 2 ATP, 2 pyruvate and 2 NADH in the whole complete glycolysis reaction. And glycolysis is a very significant reaction because it is independent of oxygen and can take place in aerob as well as in anaerobic organisms. So hence it is very crucial. And thanks. Hope you like my video. Do like, share and subscribe. Thank you.